today we're going to continue on with the isolation of gram-positive bacteria from the nose and skin. We are on part 3 this week, which is actually day 4 of the lab experiment. Okay, so there is a handout that is on Blackboard for you called part 3. And you'll see on day 4, the first thing you're doing is gathering the results from the media test that we did last week. Okay, so first of all, you're looking at the results on your MSA plate, your glucose broth, and your catalase test. Okay, so your MS plate, the results for those, okay, you should know how to read an MSA plate hopefully by now. If you don't, remember to go back to your selective and differential media lab, refer to your unknown lab, or it would be really nice if you guys were keeping notebooks like I had told you to, and it's actually in your notebook, okay, the table for the MSA plate. So you will know how to read results, okay, you will get the results for a glucose broth. Remember, this is to see if the bacteria can ferment glucose, okay, specifically ferment glucose, not ferment carbohydrates. Okay, there's lots of carbohydrates, ferment glucose. Okay, if the broth, it started out purple, remember, if it turned yellow, that means it turned acidic, and that it can ferment glucose. Okay, and then last is the catalase test. Remember, that was a glass slide with some bacteria on it. You added hydrogen peroxide. If there was bubbles, the bacteria could produce the enzyme catalase. If there were no bubbles, then it cannot produce the enzyme catalase. Okay, these results are going to be emailed to each of you individually. Okay, so you all won't get the same results. Make sure you're using the results that are emailed to you for part 3 to complete the assignment for part 3. Okay, don't use somebody else's or you're going to get it wrong. Okay, so once you get those results, from those results, you're going to be able to determine what genera of bacteria. There's a nice table on page 1 that shows you the test results and how you can interpret what genera you have. Remember, genera means the first part of the scientific name. Okay, if it's a Staphylococcus aureus, the genera is Staphylococcus. Okay, that is what you're determining from these results this week. Okay, now on to the new inoculations and tests you're doing this week. These inoculations and tests are going to tell you what the species of bacteria is. Okay, so on page 2 it tells you you are doing a blood outer plate, a nitrate test, a gelatin tube, a novobiocin test, and a coagulase test using a test by the brand name SureView. Okay, so we're going to go over how to inoculate the media and do the coagulase test quickly. Okay, so we're going to need our Bunsen burner today. Let's go ahead and get that lit. Okay, and we have our TSA cultures of isolate 1 and isolate 2, and we also have a broth culture for each isolate. I'm going to go over isolate 1, and then you would have to repeat it with isolate 2, so we'll put isolate 2's plate and broth aside, so we're only having isolate 1 in front of us. Okay, so first of all, it has you inoculate a blood agar plate. A blood agar plate has sheep blood in it, okay, and this is going to allow you to determine the hemolytic activity of the unknown bacteria, okay, meaning whether it can break down blood cells. Okay, there's sheep blood in this blood agar, not human blood. So, we're looking for the hemolysis. whether the bacteria can break down red blood cells or not. Okay, there's 3 different types of hemolysis. Okay, so you have a blood agar plate, it's all red. Okay, with blood, you're going to inoculate it, which I'll go over in a minute. But when you inoculate it, what you're going to be doing is putting a streak of bacteria. It's like you did when you did the starch agar plate. You want a nice thick area of growth just down the middle of the plate. Okay, so this will be your area of growth, for instance. Okay, and after incubation, you're looking for a halo around that area of growth, meaning those cells used the blood around them. Okay, the blood cells around them. So if it's alpha, okay, there'll be a halo around it. But the halo won't be clear. It'll actually have a greenish and olivey green cast to it. So I know this isn't olivey green, but you're still going to see partially broken down red blood cells in that halo. They'll be greenish in color. Okay? So that is alpha hemolysis. Okay? Beta hemolysis, there's complete breakdown. So the halo will be clear. There will be no blood cells left in that halo. Okay, and if you had a beta hemolysis plate, you'd actually be able to look up at the light and see right through that agar because there's no blood cells left there. So that is beta hemolysis. Gamma hemolysis is no use, no destruction of the red blood cells. Okay, so the blood agar would be exactly the same. Okay, growing right up to the culture because it had not used any of the blood cells. It'll be darker red because it's been an incubator, but you'll see the same color, red, from the blood cells right up to the growth. Okay, that's gamma. Okay, so those are the 3 types of hemolysis. All right, so to inoculate this blood agar plate, you need a sterile swab. Okay, and so we peel our swab down. Pull it out by the handle, and then we'll take our TSA plate of isolate number 1, some culture on it. Okay, you want to be able to see the culture on it because you want some heavy growth. And then you're going to pick up your blood agar plate and just like go back and forth a few times down the middle, just in that streak as in on the board. Okay, so you just have a nice line of heavy growth. Okay, so you put that aside, dispose of the swab. Okay, make sure you've labeled everything. Okay, this is blood agar, and it's isolate number 1. Okay, so that's ready to be incubated. Okay, so blood agar, check. The nitrate test, you're going to use a nitrate broth, okay, that you've labeled with isolate number 1. You're using your inoculating loop for this. It's a simple broth inoculation. Sterilize your loop. Let it cool. Pick up your broth, your plate, TSA plate of isolate number 1. Cool your loop somewhere where there's no bacteria. Get a little bacteria. Put your plate down. Pick up your nitrate broth. Take the lid off. Flame the top. Go in. Inoculate the broth. Flame the top. Now you've inoculated your nitrate broth. Make sure you sterilize your loop. Okay, so you have completed the nitrate broth inoculation. All right, next, the gelatin tube. Okay, you have done gelatin tubes before, so you will know how to read the results of it. You also know how to inoculate it, but we'll quickly go over it. Sterilize your loop. Pick up your TSA plate, cool it where there's no bacteria. Remember, you need quite a bit of bacteria again for this one, so get a clump of bacteria. Pick up your gelatin tube, flame the top, or it's a semi-solid media. Go in and stab it right into that media. Flame the top, put the lid on. You have completed inoculating your gelatin tube. Flame your loop, because remember there's bacteria on it. Okay, so you have completed the gelatin tube. Okay, a novobiocin test. So what you're doing is using the Kirby-Bauer method you learned last week to test your isolate's ability to either resist or if it's susceptible to novobiocin. Okay, so we need a Mueller-Hinton plate. Remember, we use Mueller-Hinton agar for the Kirby-Bauer method. Okay, label your plate. Remember, you have to create a lawn of bacteria on this plate to do the Kirby-Bauer method. So we're going to get a sterile swab. You'll need to use your broth of isolate number 1 for this. Remove the lid, flame the top, moisten your swab, flame the top. And then remember, you got to make a lawn, and you want a nice complete lawn. Go around the edges, go back and forth, keep turning the plate. Nice solid lawn of bacteria. Okay, make sure you discard the swab in the proper place. Okay, so these are the same cartridges you used last week, except this is novobiocin, so each of these filter discs has been soaked in novobiocin. Okay, remember you need to sterilize your forceps for this method. Okay, so we are letting this dry, lid side up so you can put the disc on the surface of the agar. Remember to sterilize forceps, you put them in the alcohol first and then in the flame. Okay, so that is dried enough. So remember, once we've sterilized our forceps, we're going to be pushing that little filter disc out so we can grab it on the other side. And then placing it in the middle of the plate, tapping it down a little bit. Remember, this might have bacteria on it, so alcohol and then flame. Okay, and now you can see that the filter disc is in the middle of the lawn of the unknown bacteria. Okay, so you're doing the Kirby-Bauer method. So that is ready for the incubator as well. Okay, and last is the coagulase test. And we are going to use a product called SureView. Okay, it's a latex test reagent. This little dropper bottle contains the test reagent. This you want to shake up really well before you use it. If you don't shake it up really well, it will not work. Okay? And you also need a little test cardboard, little circle in it they provide, and these little hockey stick applicators they provide. Okay, so a little hockey stick, hold it by its handle. You're going to need to use your TSA plate because you need to get a clump of bacteria for this. Okay, before you put the bacteria on it, you take this test reagent, put a drop of it right in the middle of that circle. Okay, this is not sterile, but that's okay because this is your last test, so it's not going to matter if you contaminate your plate. If it did matter, you want to make sure you used an inoculating loop instead and used aseptic technique. Okay, so we're just using their little applicator. Pick up your TSA plate of your isolate number 1, get a little clump of bacteria on it, and then you're going to swish it around really good in that drop of reagent. And it has to be done for probably, I'd say 10, 15, 20 seconds. Okay, you want to swish it around really, really, really good. Okay, and after you've switched it around, you dispose of your applicator in the autoclave bin. After you've swished it around really good, okay, here's your circle that's on the cardboard. So the reagent itself is a purpley blue, okay? So after you swish it around, if it's positive, it will coagulate. Okay, it will cause coagulation, meaning it produces the enzyme coagulase. And the way you can tell that is a positive will have little red droplets throughout it. Okay? And they will be obvious. Okay, so that is a positive test for coagulase using the SureView brand. Okay? A negative test, when you swish it around really good, will just stay that gray purplish blue. There will be no little droplets of red in it. That is negative. That means that bacteria cannot produce the enzyme coagulase. Okay, so then when you're done with that, you will discard that in the bin. Since that's very small and you can't see it from here, there is pictures of examples in your handouts. Make sure you refer to those to be able to determine a negative or a positive result on that. Okay, and then we will turn off our Bunsen burner, and that's it for today. Okay.